Here I'm starting out where we left off in the part one. This is part two here, and notice I'm comparing my sequence here to itself, and I have put on inverted repeats. So just to kind of show you what the parameters are, I search for direct repeats and inverted repeats, and I have maximum length of 32 and 66. Now you may have to play with this a little more to get everything you want. Remember, I am looking at the gene up here, you're looking at one down here. And I will give you the hint that your transcription start site, which is where polymerase starts copying, that would be after the TATA box, or actually I will tell you that yours is a GC box. That's going to be after it. All right, so you're going to need to find some places to start here around before 5,000. Not too far before 5,000, but before 5,000 to look for your promoter. Now I need to find my promoter up here, and how am I going to find my promoter? Well, the first thing I'm going to look for, only thing up here that kind of gives me the idea that there's something interesting going on is this little piece right here. So I want to click on that. Now it's hard to sometimes click on that. That shows the whole line. I want just that piece. And I'm not getting it. You have to maybe zoom in. You can use your scroll bar. Oops, didn't mean to do that. I think I got it there. If you can see there's just a little bit of red there, that means I actually was able to get just that region. And here is the piece. Now what do I want to do when I get that region? I want to copy it. So how do I copy this? Well, I go down here with my cursor and I just right click, and it's that easy. All right, it says select sequence region, or actually it's already selected, so I can go copy selected sequence. Now complementary, copy amino acids. Well, here we don't want amino acids, but we do want the selected sequence. So I'm going to grab that one. I'm actually probably want the complement too, but I'm going to copy just the selected sequence, which is this one here. Now we need to remember where it's at, but I'll come back to that. But here I have something already written out. I said annotation Homo sapiens RHOA. And so I'm going to paste the sequence in here, and I'm going to also put in where this was located. And this is located at 1001 to about 1000, looks like 38. So 1001, oops, to 1038. All right, so good. I've got that piece, and I'll just say forward. All right, now we want the other strand perhaps. So we could just go and find the complement, but I'm going to go ahead and do this. I'm going to go copy selected sequence, and then I'm going to go back up here. I'm going to paste it. All right, oops, it did it the same direction, not what I wanted. All right, so copy. There we go, copy selected. Let's see if that doesn't work. All right. Yes, that's what I'm looking for. So it's TAA, so that's TAA. So it's going the other direction. So this is reverse complement. All right, so we can say that this is still the same thing as above. But this is, and still forward, it's just my own notation. You can use more, but I'm going to say backward. Or I should say it's backward and it's the complement, isn't? Let's see, TAA. Okay, so that would be here's a TAA, so that would be an ATT. Yes, ATT. So it is in maybe we'd say reverse. All right, complement. And so it's in reverse and it's the complement. So I take the complement, make it go the other way. All right, so I have these two. What do I do with these two? All right, and well, I want to look for my Tata box. I don't know that I have a Tata box on this one, but I might, and that was an interesting piece, and it was in the right spot. I figure that's in the beginning of this sequence, near the beginning of the gene, probably. It probably begins in that area. We kind of have that hint because we wouldn't have been given that sequence. You may or may not have those types of hints, but I'll show you how to find those types of hints of where the gene probably begins. For us, it's kind of given to us, and I've given you 5,000 as a place to look in the sequence already, so it gives you a hint of where to look. But in my case, I'm going to have to use that little dot, that little smidge of a change there, that little line, as my first attempt to find a Tata box. Now, where would I find a Tata box? What would it look like? Now, I'm going to tell you something. Tata boxes do not have to be on any particular strand to work. They can be on either strand, and really all a Tata box is a sequence of T's and A's next to one another. Usually 2 or 3 are close to 2 and 3, and you can have some double A's and some double T's in there. So for me, what I'm going to do is I'm going to search right here, all right, for T's and A's just to see if I can see a bunch of them together. All right, and I'm going to go find. Actually, I need to do this. Now your machine may not find them quite the way it is. I need to start at the beginning here, and I'm going to go search, maybe advanced search. All right, advanced search. I want search string is TA. Searching current document. All right, and that should work. I hit search, and yes, this is what I wanted. So mine, this is why I use Notepad++. I really like it. It gives me a little bit more information. So my search results, you can see that it shows me every place I found one here, and I want TAs that are close to one another, attached, and it's because I want a TATA or something close to that. So that's one thing you can do. Now what I'm going to do is I'm going to change this a little, new search, and I'm going to put TAT, not Tata. I'm just going to put TAT. Let's see where we find that. We find that in there, but you know it's surrounded by a bunch of C's and stuff. I don't think that's it. We could do just go ahead and do Tata, T-A-T-A, but I don't think we're going to find that. We didn't. All right, so another one I might find, oops, new search, is T-A-A. Oops, go down here and see what we find. Oh, now this, we find that, and look, mostly T's and A's right here, T's and A's right here. So this region, all right, both that and in the complement, that looks promising to me. And I'm gonna close this real quick. I don't want to close it, but let's close this one. Now your notepad may not be as nice as mine, but I would say download Notepad++. It usually gives you a little bit better results, a little bit easier to use. So I like that that piece there that they were just showing us. Several T's. Where was that? Right in here. It's a TTAA TT or an AATTAA. Actually, it's a TAAT. I'm thinking that's a good place for a Tata box right there. So I'm going to make a note of that. Oops, didn't mean to bring that up. I'm going to go back to my notepad here, And I am going to notate somewhere in here, some way, somehow, that looks like a good place for a Tata box. Now, where was that? It said it was in there. Here it is. It's a nice little region like that. Now, if you're in Notepad, there's really no good way um, to highlight in these. Maybe it'll let you change the fonts. No, it won't let you change the font. I think it might let you italicize, so you might try that. But let's see what I can do in Notepad++. Can I convert the case? Yes, I can. So what I can do here is I can select it and I can make it lowercase. So that's the way I'm going to do that. And I'm going to go up here. I'm going to grab this piece and I'm going to make it lowercase because that's all I can do in Notepad. I can't. The whole idea is it would change it from a text file, and we want to keep it at this point as a text file. Now we also need to know where that is, and so we're going to have to count. So if this first one is 1001, so this is going to be 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. All right, it's going to be like around 25, 26, 27, 28, 29, 30, 31. 25 to 31. And so we kind of now have our potential, right? Potential Tata from, what was that? 1025 to 1031, something like that. I've forgotten already. So 25, 26, 27, 28, 29, 30, 31. Yeah, close enough. All right. So that would be the way we could say that. We might want to say forward because I recorded that on the forward. All right, and there we go. That's how I want you to complete finding in your case, it's going to be a GC box. So you know for your first part of this assignment that you need to find a GC box. So I just found something I think is potentially the Tata box. How would you find something that is potentially the GC box? Well, one, you're going to have to know what a GC box looks like, so I'm going to tell you. And in fact, we can just go to the internet and we're going to type in GC box, or it's CG box. And we'll type in promoter. See if we get anything. Yep, GC box right here. So you can go to Wikipedia and it's going to tell you, all right, basically what a GC box usually looks like. All right, and it tells you, hey, it may or may not come with a Tata box, actually, that is true. But here's the consensus sequence for it. It's the most likely GC box, so let's copy that real quick. I'm going to put it over here and put it back in my notepad just so you can see. If I were looking for a GC box, I would look for something that looks like that. Now, does it have to be exactly that? No. And here's what I would look for so you can get an idea. I would first take off the letters on the end. So I would first look for, because they're easier to find, G, 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 C, G. I took one off the end. All right, that'll just mark that I didn't search for that. And then I would also look for one that was not missing at the beginning, so I would go there, G, G, C, G. Okay, in another G, sorry. All right, then I would try it with the ends missing, something like this, G, G, C, G. All right. So I would begin by searching for something like this. And the idea with a GC box Okay, I'll just put this on here for you to see. Is that a GC box is a series of G's and C's at the right spot. Now, the thing is, you don't know where the right spot is exactly. Actually, I have kind of told you where the right spot is. The right spot is before 5,000. So look at 5,000, and if I were you, I would look maybe, let's give you 60. 60 base pair before 5,000. So that would be 4,940 all the way to 5,000. And in that region, I would look for something that looks like one of these. And any that you may find, you can report as potential GC boxes. And you should. Now, what you don't realize is that I've never said that there had to be only one promoter or core promoter. There can be multiple. So you need to report all sequences that might fit this kind of sequence that are, say, between 4,940 to 5,000. If you see anything that looks like that around that region, so for your test, you need to look for those sequences around, 4,940 all the way to, say, 5,000. Whoops. Look for that sequence around, here we go, and you will find one, maybe more, that will fit this parameter. Now, the thing is, you should also allow other C's in there. There's only one C in there. You should also allow any C to be substituted for a G, all right, in any of those. Now, why is that? Because the real definition of a GC box is not this. Remember, this is what we call the consensus. The consensus is, well, we looked at 1,000 of them, and this is the one that showed up most often, that sort of thing. That's not really a consensus, but that's essentially what they did. And that's not necessarily, a lot of them work that have some changes in them. Like a TATA box does not have to be TA. It just has to be a series of T's and A's clumped together at the right spot. Well, here, really all we have is a bunch of G's and C's that need to be clumped together at the right spot. And so any of these G's, there could be another C in there. It's mainly G's, a C, and some G's, but you could have some extra C's in there and it would work just fine. So you could have GC, 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 would probably work, but you need to look for those. Try to get it to be as close to this as possible. All right, it doesn't have to be identical to this, but it should be close. Now, let's go look at the GC box again. And let's say, where does it say it should be? Now, I told you, because I'm working from a TATA box, that it should be 60, but what does this say? Approximately 110. Wow. Is that, maybe we should expand our, parameters. And yes, you should. What you're going to find, I know what you're going to find. You're going to find a couple of these that are in that 60 range before there. But you're also going to find a couple that are going to be a little further away. So also look at these. The distance is not always set. The distance can change depending of where the GC box is, depending if there's also a TATA box present. So also look for one around. Now the thing is, it does not have a TATA box, but it does have a GC box. And so also look all the way back to about, let's make it 150 before. So 5,000 minus 150 was before 850. All right, and let's do that all the way up to about Let's do about 60 ahead of that, so that's 49, 10. All right, well, that only leaves about 30 in between there. So maybe you can just go ahead and look at the region between 4850 to 5000. I don't think you'll find one, just my opinion. In between there, between 49, what was it? 4910 and what, 4940? It's a small region. So answer that question when you do your report. But I'm going to tell you that those are two regions you should look in. I tell you what, yeah, that's fine. What do you find in there? Mainly A's and T's, G's and C's. What do you find in that region? What do you find? Do you find any GC boxes here? Do you find any here? So this is closer to the gene. All right. Or I'm sorry, close to the gene. The gene. This is a bit farther from the gene. Okay. And this would be the first thing you need to do on your project. Go find out, see what you can find in here, and also just look at the sequence. Now I'm going to leave this up to you. Look at these sequences and tell me if you think there is something odd going on. And what do I mean by odd? Well, is it a random sequence? Do you see a lot of something? Do you see a lot of G's and C's? Do you see a lot of A's and T's? Do you see long stretches of things? Like, do you see, 20 T's followed by 20 A's? Do you see anything odd that you can just see by your eyes and report on that? So that's what I want you to do here. Just like I did, you report these odd little sequences. Give me the forward, reverse complement. Tell me about anything potential. Now I'm finding a Tata. You're going to be finding these GC boxes. So this is kind of what you need to do first. All right, now let's go to one more thing before we end this video. Let's look at terminators. Let's see if we can find a place that would make for an interesting terminator. Now for you, it's going to be down here somewhere. Could be in this group, or it could be that little thing. I'm not going to give you any hints yet on that. Just take a look at some of those sequences. For me, I'm really thinking I should look right here. So I'm going to click on one of these, and I'm going to see what that looks like. Let's see. Sure enough, I get a bunch of A's, a G, and a bunch of A's. All right, now what does it look like? Could that bind with itself? I don't know. There's this GATGA in here. A bunch of T's over here that can bind with those. There's a gap in here. But here's some T's. Do we see anything that could pop up into a hairpin loop or anything like that? That's what we're looking for. We're looking for something where there's some A's and T's, maybe with some G's and C's in the middle. That's the easiest type of hairpin loop to find. The problem is, this is hard. This is, we have to find sequences that combine with one another and then not, they can bind with one another, but they need a piece in between them. So it's easy to see A's and T's. This very well could be, because you've got 1, 2, 3, 4 A's. Here's a fifth one with a couple of G's. So that's 4, 5. And you certainly do have 5 here in a row. The 2 G's don't worry about. Do you see some T's with a CC in them, for instance? So a bunch of T's and a CC, since these are GG's. That would be one thing to look for. I don't see anything like that. So in my particular case, this would be an interesting one to look at. I am not going to exactly find my data here. I'm going to just kind of identify interesting pieces. The actual way we're going to find the data is to take some of these pieces and run them themselves back onto on dot plot here. So we don't have to find the data on this, not the data, the terminator. I hope I haven't been saying data all the time, but terminator. I'm not going to find the terminator here today in this video, but I want to show you what you should start looking for. You want to start looking for pieces. The easiest ones are going to be when they're long sequences like this that combined with it. And actually this looks, it probably isn't. I will investigate it, but that's why it showed up here, because there are some A's and some T's, and we're getting this line going like this, and it's going there because we had some A's and T's next to each other here, and we looked for the inverse complement here. Now if I click on, instead of that, if I click on something like this, probably not, but it's worth looking at. I'm going to move up a little closer, and I'm going to look at one going this way. This one looks to me more like it could be a terminator. But we do have a worksheet, all right, that can help us out here. So just looking at those, it's hard. But we did learn that terminators, at least this example of a terminator, a hairpin loop, has this kind of sequence. Now the problem is, this is the bacterial one, not a human one. So this one is not really a good one to use, but we could look for something like this. 3 C's and ACU, but it's really not a good one to use. And the bacterial ones are a lot easier to find than the Homo sapien ones. And so I'm going to do, if it was easy as just using these, we could find it. But it's not going to be as easy because we're doing eukaryotes. So I'm going to have to make a whole new video just on terminators. This one was about finding your GC box, which you should be able to find. I will tell you right now, you should be able to find several in these 2 regions that I've given you. So this is actually the first video where you can go actually do some work. You know to look around 5,000. All right. That's the kind of thing you should be looking for. And look in these regions. Look in these regions I gave you, and that'll help you kind of narrow it down and just report on them, just like I did here. All right, I have this region. I found these potential GC boxes. Now, what do you do once you know this? Well, the only way to find out which of these is the real GC box is to do some wet lab experiments to see if RNA polymerase will stick to these sequences in the test tube. You know, in an environment where you take it and you take maybe you take this DNA sequence out and you literally just put it into the solution. You add RNA polymerase, and then you do a digest where you basically destroy any DNA that is not bound by a protein. And if you mixed it with RNA polymerase, well, that means there's going to be a protein where it's stuck. You digest everything else, and whatever is left stuck to the protein, you precipitate out the protein, and it'll still have the DNA on it, and you can then figure out what the actual sequence was, and you can figure out which ones RNA polymerase was was bound to. So that's a wet lab experiment, but you can predict that. You can predict which ones are the most likely ones ahead of time, and this is basically one way to do it. We're going to kind of predict them. So do this. This is your first part of your annotation, and I will give you terminators next time.